Magnetic resonance imaging can reveal the functional organization of our brains, even when we are simply resting, and show changes in human development and aging and disease and recovery from injury. And I'm very pleased to be here this morning to talk to you about how this works. The reason is, to use the dichotomy that Jerry Mikulski introduced yesterday, that I believe knowledge of how it works is what makes the difference between a consumer of scientific reports and a citizen with an understanding of an approach's limitations and true promise. I'd like to ask for your help. I'm going to put a request up on the screen here, and I'd like to ask you please to try to do what I'm going to ask you to do. Not for fun, but because perhaps our success at doing this or otherwise will help to inform our discussion. Okay, is everyone ready? Okay, please stop thinking. Okay, this doesn't work very well, right? But maybe that's one of the reasons that this approach does work, that we're able to perform magnetic resonance imaging of functional networks in the resting brain. So how many have had an MRI? Okay, fun, huh? Okay. So the virtue of MRI is that it's able to yield biomarkers. It's able to yield objective measurements on disease processes and, importantly, on therapy for disease, and enable us to assess this. We can, it's perhaps useful to take the rich variety of MRI methods for clinical research, so methods that seek to compare clinical populations, and put them into 4 categories based on this 2x2 matrix of whether they report on structure or function of the brain, and whether they're consistent with the principle of segregation or integration as principles of functional organization. Okay, so the principle of segregation says different parts of the brain do different things. And the principle of integration says, sure, but to get anything done, they gotta, work together. Okay? So when we're comparing clinical populations, asking questions about persons with some diagnosis versus typical healthy people, we might ask whether they differ in the size and shape of brain structures, whether they differ in the amount or extent of activation, task-related activation, whether they differ in the physical connections between different parts of the brain. These are formed by the white matter bundles in the brain, or whether they differ by the synchronous fluctuations in the resting state, which is our topic today. The task activation fMRI is more familiar in the scientific and especially the popular literature, and you've probably seen blobograms like this, right? So these are really 2 images overlaid. There's a color image of the brain lighting up, if I'm using the vernacular properly, overlaid on a grayscale anatomical image that shows the structure in the brain with exquisite resolution. Our ability to make images like this comes from the fact that MRI is fantastically flexible, sort of the opposite of a point-and-shoot camera, that just has a shutter button and no other adjustments. And so here's a simple example. These are 3 images of the same axial slice of the same person's brain on the same day in the same machine with the same blah, blah, blah. And we've simply turned a couple of knobs to differently sensitize these images. So magnetic resonance images are fundamentally images of water. Water is H2O, hydrogen atom nucleus is a proton is a little spinning magnet, and that's the source of all these signals. So this is a pretty faithful map of water concentration in the brain, of how the concentration of water varies through space. And you can see the contrast is relatively poor. It's not that easy to see different tissue types here. This is an image of signal decay. Once we create this magnetization, how long does it stay around? And here we can see the ventricles are bright because the cerebral spinal fluid, the signal maintains or persists longer than it does in tissue. And here's an image of signal recovery. Once we have destroyed this magnetization, how long does it take to come back? How long does it take for this refractory signal to come back? Now, maybe you can see these bright spots here. Can you see these a couple of bright spots? And those are due, those are due to blood vessels traversing this slice that are, and the flow of blood of bringing in fresh water, water that has not had its magnetization destroyed in this way. So that's a good introduction to the principle of blood oxygenation level dependent or bold functional MRI. The idea being that within any picture element or volume element of an image, there are blood vessels, and these blood vessels contain blood cells. And we're all familiar with the fact that arterial blood is red and venous blood is blue, right? The color, the optical properties of hemoglobin change with its oxygenation. So a red blood cell with oxyhemoglobin is red blood cells carrying deoxyhemoglobin are blue. It happens that the magnetic properties of hemoglobin also change with its oxygenation. And these blue cells here are paramagnetic, and they spoil the local uniformity of the magnetic field. Right? We spend all this money on these magnets because they don't just have strong magnetic fields, they have uniform magnetic fields. And then we do this really stupid thing. We put somebody in it, and the field is now much less uniform because of all this blue blood. So what happens when you use a part of your brain a little more? Like what happens if I tap the fingers of my hand? What's happening in my motor cortex up here that's controlling this? So you might think that the blood there would get more blue. After all, my neurons are working to do this. They're drawing oxygen out of the blood. You might think it would get more blue. In fact, the opposite happens. It gets more red. It gets less blue because the blood flow is under active control. And as soon as I start doing this, the flow of blood to that region increases in excess of what would be required to simply supply the additional oxygen. So the brain blushes. And the principle of fMRI is that we're making movies of the blushing brain. This happens to be the 20th anniversary of the first observation, which is here, one of the first 3 papers. This person is lying with the back of their head on a local receiver coil to increase the sensitivity. The first image is what a baseline image looks like. The remaining images are subtraction images showing the difference between images acquired during a certain time period and other. And when a flashing light is turned on, you can see the signal increasing. Flashing light goes off, the signal decreases, et cetera. So this was 20 years ago. And a few years after that, here's some data from our center. Similar scenario, only now we have a coil that can see the whole head just fine. Person's lying on the scanner. There's 5 minutes without visual stimulation at the beginning and at the end. And in the middle, there's 5 minutes during which a flashing checkerboard, a robust visual stimulus, is presented for on for 30 seconds, off for 30 seconds, on for 30 seconds, off. You can see the time course here of the stimuli, and we've colored in this color image are those places in the brain where the magnetic resonance signal, or the movie, if you will, the time course of this magnetic resonance movie with a time resolution of several seconds, 1 or 2 seconds is typical, matches the time course of the stimulus. So that's the key to what these images are. That is what this is. This is not a snapshot of the brain. This is a statistical map, or the colored blobs are at least a statistical map showing where in the brain does the time course of the magnetic resonance signal follow or match the time course of the sensory stimulation. Okay. Let me make this a little more formal. I was told I could include an equation as long as it's a straight line. So this is x equals g times beta plus noise. So this is just the equation of a straight line. This is the general linear model. And this is the formal way in which we ask this question, where in the brain does the time course of the signal match the time course of the stimulus? So this is space. We've taken all the 3 spatial dimensions and squished them into one. This is space, this is time. Here's the time course of 1 voxel. And here is the paradigm timing, the timing of this stimulus that you're supplying. And we have to sort of blur it out and slow it down a little so we convolve it with this estimate of the hemodynamic response function. We use regression to find the image that corresponds to the specified time course. We threshold this resulting image. And these thresholded statistical parametric maps, these are your blobs, so something like this. So I'd like to stop for a minute to congratulate you. You're now all experts on fMRI. Okay? Now, as experts in fMRI, one thing that we should be aware of is that the simple task of finger tapping activates multiple brain systems. And a neurologist can tell you what these brain systems are doing. These blobs are doing different roles. This part of your brain is basically initiating the movements. Your primary motor cortex is carrying out the movements. Your primary sensory cortex is feeling your fingertips hitting one another. And then there are these parietal regions that are monitoring the movements. And what you should know as experts, of course, is that there's nothing in the fMRI data that reports on these different roles. So our knowledge of what roles these different brain regions are playing does not come from the fMRI data. It comes from basically a century or 2 worth of neurology and electrophysiology. Okay, so the general linear model is how the vast bulk of fMRI studies are analyzed to yield these blobograms. And it's a machine that does this one thing. You specify the timing, and it tells you where there are signals that follow that timing. What if we ask a different question, though? What if we, instead of asking where do things look like I think they ought to, what if we ask what else is in my data? So there's a field called blind source separation, and the classical problem in blind source separation is the cocktail party problem. So here these 3 clip art speakers are each speaking completely independently of one another, and we have 3 microphones picking up each microphone picking up a different mixture of these 3 speakers. So the question is, the cocktail party problem is, from these 3 mixtures, or signals, can we estimate these independent sources, these original sources? Remarkably, there's a very simple answer to this that really works. It's really practical. Sorry, again, it's just a linear equation, guys. It's just, okay. Look, these are linear mixtures, right? So each microphone is recording a linear mixture of the 3 speakers. And all this is you making a linear mixture of those 3 microphone signals. And then choosing the mixture that yields the signal, that yields the sums that are maximally independent of one another. And this really works. And people are building microphone arrays into cars and using temporal independent component analysis to separate your voice from the sound of the road and the kid in the back seat. Now, auditory data are tall and fMRI data are wide, right? So auditory data have a few microphones and tens of thousands of samples per second. fMRI data have maybe hundreds of thousands of voxels and maybe just a sample every few seconds. So instead of looking for temporally independent solutions to fMRI data, we look for spatially independent solutions. And spatially independent solutions are consistent with this principle of segregation of brain function, that different parts of the brain do different things. So here's the general linear model, and remember what independent component analysis winds up being is a linear combination. Does that sound familiar? So here's the independent component analysis. See, it's just the same equation, right? Right, x equals g beta, x equals as. It's the same linear equation. The difference is here you are specifying the timing and asking where does the signal look like your timing. Here we're using higher order statistics of the signals to find sources that are maximally independent, and then whatever their time courses might be. Let me show you an example. So here we're asking the participant to fixate on this central point and alternately stimulating the left and right visual hemifields with an 8 Hertz flashing checkerboard. So you're experts, you know how to analyze these data, right, with the general linear model. You take these time courses, you convolve them with an estimate of the hemodynamic impulse response function, you use the general linear model, and you get these blobs. And for convenience, you color one red and one blue, right? So you're done. This is the community standard data analysis that you can publish in a scientific journal or maybe in the New York Times. But if we ask this question instead, what else is in our data, then things get very interesting. We find the similar activation in the left and right primary visual cortex, but somewhat larger in spatial extent, because guess what? Our estimate of what the time course wasn't quite right. And this method is a little more robust at finding these signals. We see a region with transient activation during block transitions. You turn on or off the signal in either hemifield, and this region responds. You never would have found that with the general linear model because you didn't enter this in advance. And we see activation we see synchronous activity in bilateral auditory cortex. It's got nothing to do with this paradigm, nothing to do with this task. But these regions of the brain are engaged in synchronous activity because they're functionally connected, right? So this is the insight that lets us say, well, what would happen if we were to do the same acquisition approach, only now the paradigm, instead of being flashing checkerboards or tapping fingers, is please lie still. Okay, and we acquire our bold fMRI movie just like we would do in a task activation. Well, how do we analyze this data? We can't use the general linear model because we do not have a priori ideas of what the timing should be. We can use independent component analysis, and that reveals these intrinsic connectivity networks in the human brain, networks that comport with a neurological understanding of the functional organization of the brain, showing early visual, late visual, auditory regions here. These are the where pathway, the what pathway, sensory motor activity, somatosensory, and here the default mode network that's been shown to deactivate under a wide variety of task activation and is argued to subserve things like daydreaming, which is cool. And these networks form a parcellation of the brain's cortex, sort of like taking a map of the US and dividing it up into the states. So what's going on? When we look at these networks, what is the signal in each network? Well, it basically looks like noise. If you look in any one of these networks, it's just sort of random fluctuations. But all of these regions are sharing, a same synchronous fluctuation. All of these regions are in this map, this network are sharing their fluctuations. But between any two of the maps, the fluctuations are asynchronous. The fluctuations are different. So here's an example. Here's in the motor network and the visual network. The red line is the time course in the sensory motor network, the blue line in the visual network, and the correlation is very low. These are essentially uncorrelated and asynchronous. Now let me show you some data from someone in whom that is not the case. And this is a patient with a significant recovery from what had been diagnosed as complete spinal cord injury. Someone who had a high-speed motor vehicle accident, fractured 4 cervical vertebrae, and was diagnosed with the most severe form of spinal cord injury. Many years later, this person's had a remarkable recovery, leads a very active life. He's finished not just the Ironman triathlon, but the Antarctic triathlon. Pretty remarkable individual. Now his recovery, I don't know if you can see this number, this is 47 out of 50 and 50 out of 50 for motor strength in the upper limbs and lower limbs. And then here is the sensory in the lower limb is only 9. So this person has had much better motor recovery than sensory recovery, and he reports that he uses vision a lot to guide his locomotion. He drives, and he has to look down. He has to use his eyes to see what pedal his foot is on, okay? Well, in this person, the motor system and the visual system are hugely in synchrony. So this fellow who's using vision to guide locomotion, using vision to guide his feet, when he's just lying on the scanner, please lie still, his visual and motor systems are highly synchronous. So I think this finding is significant because it reports upon the brain plasticity that underlay his recovery. And we're now working to look at additional patients at the International Center for Spinal Cord Injury and try to see whether this kind of study can help to guide activity-based therapy to help people recovering function after injury. The final time I'm going to show you this one linear equation, this time with seed-based temporal correlation. This is a different approach. We can pick a location on the brain and say, hey, what else in the brain has a time course that looks like that region? So it's sort of like the general linear model, only instead of taking a time course a priori, you're taking a time course in some region. Or we can also look at seeds. So here are 3 of the prime loci in this default mode network, and this shows how highly synchronous they are. Again, each one just looks like noise, but gosh, it's kind of the same noise in each of the 3 seeds, which are part of the same network. Let me show you some people in whom this is not the case. And these are individuals in the pre-symptomatic stage of Huntington's disease. So these are people who carry the mutation in the Huntington gene, but who are perhaps decades away from a clinical diagnosis, decades away from any clinical motor symptoms. And in these individuals, there is reduced synchrony in this default mode network years before they would have clinical symptoms. And I think this finding is significant because it offers the opportunity to use these kind of results as a biomarker for therapy. I mean, this is when you want to do therapy, right, is before the symptoms appear. I just want to close. I want to ask about individual differences. So let's say, these resting state signals in your brain are different from these resting state signals in my brain. Does that really mean anything, or is it just some sort of technical glitch? Well, children vary quite a bit in motor control. And motor development is something that increases during human development. So this is a cross-sectional study, not a longitudinal study, but a cross-sectional study. Each dot here represents a child. These are typically developing children, all right-handed, between ages 8 and 12. This is their motor control based on a clinical examination. And this is, if you will, the handedness of their connectivity, the difference between functional connectivity in a left hemisphere and right hemisphere brain network, motor network. And this is a strong correlation. So this association is neat. I mean, it's neat for us who are doing the technology because it suggests that the stuff we're measuring is really relevant to who you are and how you do, and it may have some significance as we look across the population. Indeed, the question of individual differences is there's so much enthusiasm about this that we recently put out, this is the number of subjects, not the number of authors. We contributed data to what's called the 1,000 Functional Connectomes Project, sometimes called KFC, which put out on the web, where anybody in the world can download them and analyze them, such resting state fMRI data for more than 1,000 individuals. And here you can see from that paper some brain regions, parts of the default network, that are showing increases with age and decreases with age. So I think this paper especially, I think, shows the enthusiasm and the interest and the popularity of this approach to looking at functional connectivity in the resting brain, which removes basically requirements for compliance. So we can apply this approach to clinical populations that would have difficulty carrying out a variety of tasks that might be required for fMRI. Think of people with spinal cord injury who are paralyzed, who would be unable to comply with requests to make various motions. But we're able to see these motor control networks in the resting state. I think this is all a very happy outcome from the observation that Pascal made a while back, that we're unable to sit quietly in a room alone. With that, I want to thank my collaborators and funding agencies, especially all the research participants, and thank you for your attention.